All right. Yeah, no, we're on the C3 cycle. All right. So we're on the C3 cycle. That's what we started before the exam. So that's what we're picking up on now. And I'm glad to see that you're made it through hopefully plowed roads by now and power back on after the weird storm. All right, so I wanted to start here. This is, I think, pretty much where we left off. We were looking at the so-called pie chart diagram of how plants grab CO2 from the air, bring it in through their stomata, and then start working these reactions to get their sugars. And so carboxylation, we covered that in pretty good detail last Wednesday. Geez, a whole week ago. And then we saw how that reaction does not require any ATP, does not require any DPH. So this is the carboxylation, where we have CO2 coming in, reacting with that RUBP. So we call the RUBP molecule, ribulose 1,5-bisphosphate, it's often referred to as an acceptor molecule because it's accepting CO2 as it's coming into the plant. And so RUBP and CO2 react. That's called a carboxylation reaction. And you get this temporary unstable 6-carbon, and then it breaks into 2 identical phosphoglycerate molecules. The 3 just means that it's the 3rd carbon that has the phosphate. So that's what's going on during the carboxylation step, and you see there's no ATP or any DPH. So this will happen. Yes, 2 of them. 2 identical PGA molecules. And so that gets us to this area here, which is 3-phosphoglycerate. So there's 2 of those that are made for each RUBP that accepts the CO2 and reacts with it. So now we have to work through the next step, which is the reduction phase. And so that means we're going to be adding electrons to PGA in this reaction. And you see that there's both ATP and NADPH involved. So NADPH is the molecule coming from the light reactions. NADPH is very happy, chemically speaking, to donate electrons off. And that's now we finally see its role. It's going to come in and donate electrons, and then ATP is going to be involved to push that reaction. So let's look at this. The reduction phase is, yes, you have to use a little bit of energy input, ATP. You need that reducing molecule, NADPH. And then you will be able to work that reaction until you get PGA, or to reduce PGA into G3P. So what's going to happen here, you don't need to know this in detail if you just know that PGA is going to glyceraldehyde 3-phosphate, which you can abbreviate to G3P. You don't have to write it all out. So PGA to G3P, there is an intermediate. You don't even know that word, or the intermediate. So you go from PGA to G3P. And please get this straight in your head. I've seen students mix these up in previous semesters where they'll mix it up. So I know it's a little, they're both 3-letter words or abbreviations. So let's make sure that you're getting this straight from the get-go. So we have G3P, often it's referred to as triose phosphate. Triose just means 3 carbons. And what we're going to see is this is kind of a branch point in the stage of the carbon fixation. Some of the G3P is actually going to get sent out of the chloroplast and into the cytoplasm. Most of it stays in the chloroplast, however, right? So we'll talk about that. When it is getting exported, it actually gets rearranged into DHAP, dihydroxyacetone phosphate. So you can just abbreviate that down to DHAP. So G3P rearranges to DHAP and then boom, gets sent out into the cytoplasm. And that's where you get sugar production. So we're going to hold that thought. We're going to stick to staying in the chloroplast and finish up the Calvin cycle before we think about sugar production. And so what we see is that most of the G3P that does get synthesized, it does stay in the chloroplast because we need more RUBP to be made. So this is a, a cycle, Calvin cycle. So we need to get some RUBP up and ready to go to be ready to take in and accept more CO2 that's coming into the plant. Okay, so let's look at this in chemical means. So here's your PGA on the far left, and you can see that it gets phosphorylated and then it gets reduced. And so that final product is G3P. So PGA to G3P. Now that G3P, we're going to see we need to get enough of those put together to make more RUBP. RUBP is 5 carbons. G3P is only 3 carbons. So you can tell we're going to have some reactions to think about in the regeneration phase so that we can make more of that 5 carbon RUBP. So in order for the Calvin cycle to keep going all day long, as soon as the sun comes up and it strikes the plant, that will stimulate them to open up their stomata and they'll take in the CO2. So all day long, they want to be making sugars. And so you need enough RUBP to let that continue over and over and over and over. And so the regeneration phase is getting this idea met where you have that G3P and it's going to get converted into RUBP. So remember, we call the RUBP the acceptor molecule because it's accepting CO2, it's reacting with CO2. Now, on the pie chart, the regeneration is the last step of, first you have carboxylation, then you have reduction, now we're into regeneration. It's the third phase of that pie chart. There's actually a lot of reactions that are going on during this phase. So I'm going to tell you what you need to know in detail and what you can just gloss over. And so it's a series of reactions, and we're going to see that the reactions involve 4, 5, 6, and 7 carbon sugars coming together and reacting, and it's really kind of like playing a game. You got to end up with 5 carbons. You want 5 carbon RUBP at the end of all of this. And so we're going to see as they're combining G3Ps and making these different carbon compounds, the goal is to get to 5. That's the magic number. So I like to call it the numbers game for this step. And so when you look at it and you're figuring it all out, you're saying, what's going on here? The net effect is that if you start with 6 G3P molecules, 5 of those are going to get used to make 3 RUBP. Okay. So that's the big picture idea of what's going on in the regeneration. So as you can see, 5 of every 6 G3Ps. So only 1, in essence, what that's saying is 1 G3P gets exported of those 6, 5 stay in the Calvin cycle because they're necessary to have enough carbon compounds to end up with 3 RUBP. Okay, so regeneration on this pie chart, you can see it's, the tan part of the pie chart here. And you can see that there will be some input of ATP in key steps to get this to completion. So you can see that both in the reduction and regeneration, we do have ATP coming from the light reactions. And then the reduction phase, you have that NADPH coming from the light reactions. So that's how they're linking together. Okay, so your book has, I put in a couple of different figures. So this one, I believe, is in the current book that you're using. And I left this in the slide set because some of you work really well with the chemistry and looking at the chemical compounds and following the reactions that way. If this helps you, fine. It doesn't help me. I don't prefer learning it off of this myself. So I don't really use this, but if this works with your brain, great. Then there's this one that's from an older book, And again, it may help you, so I put it in there. So these 3 slides in a row, this one's in the middle, this one's in the middle of these 3 slides I just showed you. This is the one I like to work off. It's pared down and my brain just works better. So this is what I teach off of. It's a little bit simpler in looking at how these G3P molecules are combining. So I want you to play the numbers game as you're learning this, not spelling out and memorizing all the words of the chemicals, that's of the molecules. I'm not asking for that. I'm asking for you to know how to play the numbers game. Okay, so what does that mean? Well, we have the G3P, so we often refer to this as the G3P pool because there's 6 of them there. So it's a pool of these molecules waiting to get into the regeneration. So what do we have happening? So these are 3 carbon molecules. So you have 2 of these G3Ps, 3 carbons and 3 carbons, they react to give you a 6 carbon fructose 1,6 bisphosphate. All right, so those 2 react, and so you end up with a 6 carbon. Then what you can see is that fructose 6 phosphate, that 6 carbon, is going to react with another G3P. Okay, so right here you see fructose and another G3P reacting. So 6 plus 3, that's a 9 carbon. They don't really show the 9 carbon molecule here where the red laser pointer is. You have to imagine a 9 carbon that quickly splits into a 4 and a 5. So here we have a 5 carbon molecule. That's good. We're getting close. Remember, RUBP is 5 carbons, so that's pretty good. So we're going to leave off on that one and just look at this 4 carbon for a second. So we need to somehow get to a 5 carbon molecule with this. So what you could do is you see another G3P coming in and reacting with that 4 carbon. So that gives you 7, the cetoheptulose 1,7 bisphosphate, that's 7 carbons. That's going to react with another 3 carbon, so 7 plus 3, now you're up to 10. And if you divide that by 2, you get 2 5 carbon molecules. So by the end of this, if you follow these arrows, you have a ribose 5-phosphate, a xylulose 5-phosphate, and a xylulose 5-phosphate. So those are all 5 carbons. They rearrange to ribulose 5-phosphate, and then they get phosphorylated because this has 1 phosphate, the ribulose 5-phosphate, but RUBP has 2 phosphates. So now you can finally see where the ATP needs to be put in to this reaction so that you can get the full RUBP regenerated. Okay, so this is, if you like the words of the molecules, the names of the molecules, they do give you a little bit of clues of how many carbons you have, but really if you just, recognize that G3P is 3 carbons and start working it, you will be able to follow the carbon counting. So I'm not asking you to memorize the words. I'm asking you to think about the numbers games of how do you start with these 5 G3Ps and end up with ribulose 5-phosphate. This is the only reaction right here that I want you to know in detail. Ribulose 5-phosphate with ATP plus ATP, it has to come in so that you can get RUBP. So this last reaction, I need you to know the words and that ATP is involved to get you RUBP. All the stuff leading up to it, you certainly have to be able to tell me that you understand this. G3P is 3 carbons. Okay.
So would you ask something like, okay, after step 5, how many carbons would you have? Is that something?
What do you mean by step 5?
Right there, like step 5, like after 3.
No. Okay. So what I'm asking for is you understand how you have these 3 carbon molecules. How do we end up with these ribulose 5-phosphate? Let me see if it's in here.
Because in the previous class we had to label how many carbons were in. That's why.
Oh, no. So let me pull up I'll, I don't know if this is showing up in the Zoom, so let me do this again. Oh, it is not behaving. Hang on. All right, let me try sharing again. Oh boy. Wow. Let's try this. All right, I'm not sure if this is going to show up in the Zoom or not, but we're just going to go with it. Okay, so let me, oh, come on. Okay, so here is, all right, so this, let's go to this again. All right, so what I would probably envision asking you on the exam would be something like this, and I'll post this into EDUCAT. So as long as you understand that the 3 carbons, that's your G3P, you need to define that for me so I know that you know. But basically it's this kind of idea. You don't need to write out the words, you don't have to know every enzyme, all right? So it's 2 of these G3Ps reacting that gets you a 6 carbon. And then another G3P comes along and reacts with that, so then you have a 9 carbon. That 9 carbon breaks into a 5 carbon and a 4 carbon. So I've circled the 5 carbon in green. That's like, okay, we got the 5 carbons. We're good on that. We're close. So I'm going to hold off on that and just now work with the 4C. That's why it's an asterisk, so that you understand that this 4C asterisk is the same as this 4C asterisk. So that 4 carbon is going to react with a 3 carbon and get you 7 carbons. That's going to react with 1 more G3P, and that gives you 10. And then if you break those into 2 5 carbons, now you see the 3 circled 5 carbons. Those can rearrange and they all become ribulose 5-phosphate. So now you have 3 of these ribulose 5-phosphate. So now we're really close. Not quite there because we have to add in ATP to get the ribulose bisphosphate, the 3 of those now. So this is basically what I'm looking for. You don't have to know names or enzymes. It's the numbers game. Questions? So what we just worked through right here is being shown here. It's just that they put in the words and added a little bit of detail. So these 3 5 carbons, there's a xylose 5, a xylose 5, ribose 5. They're all 5 carbons. They rearrange to ribulose 5. There's 3 of them. And then 3 ATP come in and react and end up in 3 RUBP. So this is if you have 3 CO2 and 3 RUBP working in to get to 6. That's usually how we talk about it because it's just easy to play the numbers game. If you work it out where you can end up with 6 G3P to talk about, so that it's much easier than to think about the regeneration. That's really the only reason they do it this way. It's just it works out, the number game works out beautifully if you start with those pool of 6 G3P. So I'll post this. So what this is showing on this black and white figure here is the whole thing. The whole pie chart we were just looking at. It's just laid out in a little bit more detail where you can see here at the very top left, you have the 3 CO2 and 3 RUBP reacting. You get the PGA at the end of that, so that's carboxylation. And then PGA gets phosphorylated and reduced. You get your G3P, that's reduction phase. And then the G3P, the vast majority of this black and white slide, that G3P pool, all of this coming back to ribulose 5-phosphate. That's all regeneration. So definitely regeneration is the biggest component of the 3 steps. Very important because you got to get that RUBP all day long. Plants have to keep running this. And so if they run low on RUBP as CO2 is coming in through the stomata, they got nothing to react with. That's terrible for the plant. You want to keep the Calvin cycle cooking along every single minute that the sun is shining. Plants are taking in the CO2, running the light reactions, making sure there's enough NADPH, enough ATP to get this going as well. The Calvin cycle needs that NADPH and the ATP. Okay. So the regeneration, let's go back. Let me share again, see if this is going to behave. All right, so the regeneration phase, definitely get that RUBP, enough of that made so that all day long, as the CO2 is coming in, you can get the sugar. So here, what we talked about is this pool of G3P, and you see here, you can follow down here, there's a couple of options. So here they're showing toward some of the G3P. So this very last one, the artist decided to pick on the last G3P in this picture and say this is the G3P that's destined to go out into the cytoplasm. So export means it's coming out of the chloroplast, getting into the cytoplasm. Some people call it the cytosol. This DHAP is how it gets exported, the dihydroxyacetone phosphate. So that will go out into the cytoplasm and you'll get sugars made. Here, these other 2 G3Ps that you see, they combining and you get that fructose 6-phosphate. They say sometimes the plant will make the decision, so to speak, to push this all the way to starch production. And so if the plant has lots of sugars, it's got plenty to keep the Calvin cycle running, it may siphon off some, so to speak, in order to make starch, which is their long-term way of storing carbon. And so they'll pack it into starch granules, and you can actually see it inside the chloroplast. You can see the starch grains starting to build up in the chloroplast. So starch is an option. Sucrose, certainly, they have to do that as well. Plus, keep the Calvin cycle going so that you can keep enough RUBP up and running.
So would this be, would that be like certain changes? Would that be instead of the G3P going to RUBP or
in addition, the instead word comes into play here, where when a plant is deciding, should I make sucrose or should I make starch? That is an either or, one or the other. They don't do both in the same chloroplast. And so, and we'll talk a little bit about that. And so the plant will export some for sucrose, and then the other option is starch, and it's an either or idea. But in the meantime, they're keeping the Calvin cycle running. As
different G3P.
Yeah, exactly. So some of it gets sent off into sugar or starch, but you have plenty of other G3P to keep rolling into the regeneration phase to get more RUBP. You don't want RUBP levels down low during the day. You want that really lots of RUBP so that the CO2 has something to react with when they come in, when it comes into the plant.
Could that be like before camp build it up overnight or something?
Yeah, we'll talk a little, actually, we're going to talk about this idea of RUBP. So at night, this isn't happening at night. Okay, and we'll talk about why that is, but just for now, it's not happening. So you can imagine overnight, the RUBP levels could drop pretty considerably. And so they looked into that and they did figure out that plants have a way to deal with that issue, because as soon as the sun comes up in the morning and strikes the leaves, they want to have RUBP up and ready to go. They don't want to waste a single second of sunshine. And so they have looked at how do plants regulate the C3 cycle to make sure that it's optimized for photosynthesis during the day. And so there's this idea that they've termed autocatalysis. Basically what they're saying is at night, the Calvin cycle starts to slow way down and eventually, kind of just stop. And so what they're finding is that you have extra carbon in that the plant has available. It gets diverted to making RUBP. And basically the big picture idea of this is that they have a way to regulate to make sure that RUBP levels don't crash overnight. That's the big picture idea I want you to grasp with this. And so in the morning, as the sun strikes the plant, if they did everything correctly through the night, they have enough RUBP ready to go. And then of course, as the sun strikes the plant, they'll continue to make RUBP all day long. But at night, they run the risk of RUBP levels dropping, but they have figured out a way to regulate their carbon compounds at night to make sure that there's enough RUBP in the morning. Excuse me. Okay. So what's important to realize is that we say that photosynthesis doesn't take place during the night. And so the carbon fixation, making sugars, they can't really do that during the night. And the reason that is that there are enzymes involved in the Calvin cycle that need light in order to be active. And if they don't have the light, they just kind of go to sleep and won't function. So far, they've discovered 5 enzymes. One of them is Rubisco. So Rubisco is fussy. It needs light in order to function. And then there's 4 other enzymes involved in the Calvin cycle. I'm not going to ask you to memorize the names. Just know there's 4 others in addition to Rubisco. And they're still working on this. They're still trying to piece together if there's more enzymes than this. So far, they know of 5. So the 4 others that are on this list, the 4 others besides Rubisco, what they've discovered is they all have the same mechanism where they become active with sunlight. They have sulfur-sulfur bonds and without light. In the presence of light, they end up with a situation where those sulfur bonds get broken. And that is the stimulus to get that enzyme active. So the S to S bond, they're inactive. When light kicks into the system, they call it the ferredoxin thioreducin system. Nice big mouthful. Ferredoxin thioreducin system. Basically it's a short little redox reaction where you have electrons being donated and accepted, and I'll show you a picture of this. And ferredoxin is one of the major players. So we've already learned about ferredoxin in the light reactions. All right, so the light reactions, you do see ferredoxin getting an electron in that process. So now here we see a situation where ferredoxin can kick this into gear. So here it's ferredoxin. You can go back to the light reaction. You see that ferredoxin is associated with photosystem 1, waiting for that electron. Once it gets the electron, it releases off, moves into the, the main portion of the stroma. And if everything's going okay for the light reactions, it'll bump into FNR and get you your NADPH. But ferredoxin can also take an electron that it grabs from photosystem 1, and it can enter into this light activation redox reaction. So here is the idea. So what they're showing here is a short redox reaction. So the reason they say light is needed is because in order for ferredoxin to get reduced, the light reactions have to kick into gear. Otherwise, it's not going to happen. So that's why they have it here with the chlorophyll and the light. They're saying you have to have the light reactions. You have to have light. That will get you a reduced ferredoxin. And then what happens is this ferredoxin donates an electron to this second enzyme. So you can see the sulfur-sulfur. When this enzyme accepts the electrons, so these two react with each other, the electron, and that breaks the bond, the sulfur bond. So now this has gained an electron. Now it passes it to the second enzyme that also has a sulfur-sulfur. So when this one gets it, breaks the bond. And then again, so where they say enzyme, they're saying that's one of the 4 enzymes that they've discovered in the Calvin cycle. So now this enzyme, this TRX enzyme, gives it to one of those 4 enzymes, and again breaks the sulfur bond. Now it's active. So it's a short electron transfer. Ferredoxin kicks it up, and the only way you can get reduced ferredoxin is with light. So that's why they're saying those 4 enzymes need light in order to be active. So therefore, the Calvin cycle does not happen in the dark at nighttime. They just stop. They stop doing it.
They are
Yes. So it's an electron transfer from ferredoxin to a couple of other players, and then finally one of those 4 enzymes. This is also from a different version of your textbook. I put that in. It's saying the same thing. It just, if it helps you study this, I just added it in. And then this one too is saying the same thing. It's just slightly different visual, where you have the idea of photosystem 1 getting hit with light energy and gets excited, and eventually you get a reduced ferredoxin to the light reaction. So this is slightly different visual. If it helps, go ahead and use it. But these 3 slides are basically saying the same thing. Okay. So those 4 enzymes, that's what they do. They work through reduced ferredoxin, will kick off this very short electron transfer reaction, redox reaction, to get one of those, each of those 4 enzymes active. Rubisco is also light activated, but it's a different mechanism. It's not that ferredoxin thioreducin system. We're going to see that now there's a second carbon dioxide that plays a role to activate Rubisco. So this is not the same CO2 that reacts with RUBP. This is different. So there's a so-called activator CO2. That's not the CO2 that binds with RUBP. It's a different one. It comes into a portion of the active site. It's an amino molecule, the lysine amino acid there. And they call that a carbamate form of Rubisco. Once that activator CO2 has come in and bound there, they call that the carbamate form of Rubisco. So we're close, but not quite. So then once that CO2 has come in, that activator CO2 has come in, the next thing that has to happen is magnesium comes in and binds with that activator CO2. So you have Rubisco, you have the activator CO2 coming in, and you have magnesium binding to that. Once you have that, then it's complete. Rubisco's been light activated. So, you know, looking at what I have here on this slide, there's like, okay, where's the light? I don't see, what do you mean by light activated? What's going on? So let's put this into the context of how this needs light in order for this to happen. So here, you see Rubisco up top. Here's that lysine residue, and you can see that you have CO2 coming in. So that's the carbamylation reaction. I don't know why every time I hear carbamylation, I always think of ShamWow commercials. I don't know why. but I do carbamylation. Isn't that ShamWow, right? The ultra absorbent tunnel. Anyway, so then the magnesium comes in and binds to that carbon dioxide. So now you see at the very end, Rubisco is active. So what's going on with the light? I said that involves light. So what I've tried to draw here is that in the chloroplast, so remember, this is happening in the stroma, okay? So light energy is striking the chloroplast, and as the light reactions are cooking along, one of the big ideas is that you get a proton gradient, right? You get protons getting dumped into the lumen. If you get enough protons dumped into the lumen, that's going to drive your pH slightly acidic. So that's why I put pH 5, because when you measure pH, it's measuring the concentration of protons. So as the protons are building up, it gets more acidic. That causes a situation where magnesium flows out of the lumen and goes into the stroma and then can attach to that activator CO2. So to get Rubisco fully active, it needs that magnesium. And the only way for that to happen is if you have the light reactions cooking along to create this acidic situation where proton comes in, magnesium goes out. And if you get that situation, then the magnesium can come out and bind with that activator CO2. So that's why they say Rubisco has to have light in order to be fully active and functioning, because Rubisco has to have that activator CO2 and the magnesium attached to it. This is the same idea. So whatever version you want to stare at when you're looking at it and convincing yourself of the situation, it's fine. Okay, so Rubisco is the most abundant protein on the face of the earth. And so they've been heavily studying Rubisco and how it gets regulated. And so this was a big, huge deal when they discovered this whole process. So remember that CO2, that's the activator CO2. That is not the CO2 that's reacting with RUBP. They don't even show that happening right here. They're just showing how Rubisco is getting activated. Another thing that they've discovered is this enzyme, Rubisco activase. So it's activating Rubisco. It's helping to activate Rubisco. So what's happening is during the night when the Calvin cycle stops running, one thing that they see happening when that's all happening at night is that Rubisco gets a bunch of sugar phosphates attached to it. I often think of it as a blanket of sugar phosphates covering Rubisco so it can take a nap during the night. So at night, you have those sugar phosphates. Well, so that's what makes Rubisco go quiet during the night. But as soon as the sun rises, you want Rubisco to take the blanket off, right? You need to get up in the morning, get the blanket off, get Rubisco activated. And so what happens is the Rubisco activase will come along in the wee hours of the morning and start getting rid of the sugar phosphates. And that helps Rubisco get ready to go to get activated. So as soon as the sun strikes the leaves, it can start taking in CO2. Some of the CO2 will react with RUBP. Some of that CO2 starts to activate Rubisco with the activator CO2 and the magnesium. Okay, so Rubisco activase is a very important enzyme to kind of get things going, take the blanket off, so to speak, of the blanket of sugar phosphates off in the wee hours of the morning so that Rubisco can get fully activated.
So what the CO2 just not know how to find the activation.
Yeah, so if there's sugar phosphates in the way, the activator CO2 can't physically get in there and do its job. Okay. So there are other ways in which Rubisco has found to be regulated, but that's enough. I'm not going to keep going on in that, but just be aware that, Rubisco is incredibly important. It makes sense that it is pretty tightly regulated by the plant through a variety of mechanisms to make sure it's nicely optimized so that you have this Rubisco enzyme helping out with that very, very first step of the Calvin reaction, the Calvin cycle. Okay, Rubisco is interesting though, because that enzyme has, so the C in Rubisco stands for carboxylation, which is what we've been talking about. The O, on the other hand, stands for oxygenation, and that's a whole nother ballgame. So now Rubisco has this funky ability. It's not very stringent. If CO2 comes into its active site, it'll say, yeah, great, I'll react CO2 with RUBP. However, if oxygen comes into its active site, instead of saying, no, oxygen not allowed, it says, oh, all right, I'll take oxygen. And then oxygen reacts with RUBP. And that's not the Calvin cycle. Instead, it's called, well, some people call it the C2 oxidative photosynthetic carbon cycle. It's also called the photosynthetic carbon oxidation cycle, which nicely abbreviates down to PCO, which is what I prefer to call it, because I like the PCO word. I don't like to keep saying photosynthetic carbon oxidation cycle. So PCO cycle, some people call it photorespiration. And I'll try to explain why they call it that as we work through this. But the upshot is Rubisco is not very stringent. It doesn't have a huge high binding affinity just for CO2. It's a little sloppy. And so it can take CO2 into its active site, but it can also take O2 into its active site. So it just depends what comes into its active site, if it's going to be a carboxylating enzyme or an oxygenating enzyme. So we say that CO2 and O2 compete. Basically, Rubisco is saying whatever there's more of around me, that's what's going to come in, just by the laws of, diffusion. Higher levels of CO2 around Rubisco, then CO2 comes in. If there's a situation for some reason that CO2 levels are really low and there's plenty of oxygen around Rubisco, then oxygen wins and comes in to the active site. So it's a full competition idea. Whatever there's more of around Rubisco, that's what gets brought in to the active site. Well, if oxygen is coming in, what we see is this reaction. So Rubisco says, oh, there's more oxygen around. I'll take oxygen. I'll react it with RUBP. And in that reaction, you do end up with one PGA molecule, not two, just one. And then you get this funky 2-carbon molecule called phosphoglycolate. So remember, doing the carbon counting, you look at the substrates. RUBP is 5 carbons. O2 has no carbons. So you end up with a 3-carbon PGA. That only means you have 2 left over. So it's a 2-carbon phosphoglycolate. Well, that's good and bad. The PGA, that can feed into the Calvin cycle. So that's good. At least one of the products can get fed directly into the Calvin cycle. But that phosphoglycolate can't directly feed in to the Calvin cycle. There's no enzyme in the Calvin cycle that can grab it and easily incorporate it into the Calvin cycle. So what we see is that the phosphoglycolate, instead of going into the Calvin cycle, the C3 cycle, it shifts into this thing we call the PCO cycle. So if you look at the PCO cycle and you follow it from start to finish and say, okay, what's going on here? The upshot is that you do get some partial recovery of carbon. What they mean is you're able to actually make some more PGA through this PCO cycle. You do recover some PGA from it, but it takes a little effort, as you'll see. But in that process, we say that some carbon is lost. What we mean by that is it's not being used to make PGA for the Calvin cycle. It's actually getting respired off as CO2 over in the mitochondria, and it's like given off by the plant. So it's respired, so that's why they call it photorespiration. So this is generally not a good idea. So when you look at the PCO cycle, it's complicated. You lose carbon as CO2, extra energy is needed to push it. So when they do experiments in the lab and they create a situation where they make a plant run the PCO cycle, they force it, like run the PCO cycle over and over and over. What they see is that this plant doesn't do as well as a plant that barely runs the PCO cycle. Their biomass is less, they're more susceptible to issues. So in general, it's not a good idea for a plant to run the PCO cycle, especially on a regular basis. If it happens every now and then, yeah, but on a regular basis, no. And so the PCO cycle is not really what a plant should be doing on a regular basis. This is trying to put in a visual what can happen with Rubisco, whether it takes CO2 or O2. So on the left in the peach colored circle, that's the good one. If RBP takes CO2, you get the 2 molecule of PGA, and things cook along with the Kelvin cycle. Great. On the gray end of this, you see oxygen coming in, and you get a PGA, but you also get that funky glycolate. You give off CO2. Not good. If you want the visual in the chemical reaction, this is what's happening. So you have the REBP reacting with oxygen that only gets you 1 PGA. And then that funky 2-carbon phosphoglycolate. Not good. So now that phosphoglycolate has to enter into the cycle. And it's complicated. So let's see. This I put in showing the carboxylation reading up and then the oxidation down. I'm not really spending time on this. It's just a visual again for you if you want to look at it that way. Okay, so this is the PCO cycle. This is also the PCO cycle that your book has. I don't like this version. To me, it's too messy to learn off of, in my opinion. So I don't teach off this, but it does give a lot of detail if you need that as you're working through it. So I'm going to teach off this one. Okay, so the upshot is here. The PCO cycle is complicated. It has 3 different organelles involved in it. It kicks off in the chloroplast. So we are looking at the top left here for the start, where REBP reacts with oxygen instead of CO2. So you see, you do get 1 phosphoglycerate that can enter into the C3 cycle. Then you get that 2-carbon phosphoglycolate, and you can see now that has to go into the peroxisome, series of reactions, go into the mitochondria, series of reactions, come back to the peroxisome, come back to the chloroplast, and finally you get 1 more PGA through all that mess. Yeah. So next time we're going to talk about the PCO cycle, and I'll make it clear what I expect from you. We're not going to get into the details of this, but there's a couple of key things I want you to grasp. Okay, so we'll start that on Friday.